Okay, we're going to make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we're going to summarize everything that we have looked over in the last couple of weeks. We have looked over all of nucleic acids, so DNA replication, structure of DNA, RNA, ATP, water, and inorganic ions. We're now going to just summarize by looking at some exam questions in today's lesson, and then in our next lesson, we are going to be moving on to our new unit, which is all about cells. Okay. At any point, if you have any questions, you can let me know in the Q&A, and I'll answer them as soon as I can. And then if you want to attempt these questions, you can attempt them at any point as well. Okay, so our first exam question is 6 marks, and it says outline the similarities in and the differences between the DNA and RNA molecules. Okay, so we looked at DNA and we looked at RNA, and we saw that both of these were polymers, however, they have different structures. Okay, so both of these are polymers, so we can say that they are polynucleotide strands, or they are both a polymer of nucleotides. We can say anything like that. And then we're going to dive into the structure. And because it says similarities and differences, we need to keep an even split when we're talking about our answer between the similarities and the differences. Okay, so all I'm going to do now is I'm going to write a subheading similarities. Some people like to draw it as a table. Just because of space, I'm going to draw as a table as well. Okay, so we've got similarities and we've got differences. Let's start off with the similarities. Both of them are a polynucleotide strand, which means that they are made up of many repeating units. Okay, so both of them are a polymer of nucleotides. And remember when we looked at this before, I said whenever we have a polymer of nucleotides, instead of writing polymer of nucleotides, you can write one word, and that is polynucleotide. Okay, and that means the exact same thing there. Okay, so we're starting off with the similarities. Now, the next thing is let's dive into the structure. When we look at DNA, we know that we have a pentose sugar, we have a nitrogenous base, and we have a phosphate group. Okay, and that's the same within RNA. Okay, RNA also has a pentose sugar, it has a nitrogenous base, and it has a phosphate group. Okay, so our similarity is both have pentose sugar, um, pentose sugar, nitrogenous base, and phosphate group. Okay, now I would say one more similarities. Let's look at the bases. Okay, we know that both DNA and RNA have similar bases. There's only one base that's different. Now, the bases are the same. We're just going to mention them. Both of them have cytosine, guanine, and adenine. Remember, the thymine is not present in RNA. The thymine turns into uracil. Okay, so we're just going to mention that both have cytosine, adenine, and guanine as the bases. Okay, that's what we're going to say. Now we've stated 3 similarities. I'm going to state 3 differences now. And one of the differences is going to build up from the similarity. We said that both have a pentose sugar. In RNA, the pentose sugar is ribose, and in DNA, D for DNA and D for deoxyribose. So the difference is that DNA has deoxyribose, while RNA has ribose. Okay. And my second difference is also going to build up from a similarity. We said that they have cytosine, adenine, and guanine. Well, DNA has thymine and RNA has uracil. Okay, so we know that DNA has thymine, whereas RNA has uracil. Okay, and then one final difference, the key difference between RNA and DNA is that one is double-stranded, while the other one is single-stranded. So DNA is double-stranded, while RNA is single-stranded. Okay, so that's how I would compare the structures of DNA and RNA against each other. Okay, so it's 6 marks. So ideally you want 3 similarities and then you want 3 differences. In terms of the similarities, we know that both of them are nucleotide strands. They're repeating units, so polynucleotides. We know that both of them have a pentose sugar, a nitrogenous base, and a phosphate group. And we know that both of them, in terms of the nitrogenous bases that they have an option of cytosine, adenine, and guanine. Now, the differences were made a bit easier because they stand out from the similarities. We can use the similarities to work out the differences. We said they have this. They both have a pentose sugar, but the difference is DNA has deoxyribose as the pentose sugar, while RNA has just ribose as the pentose sugar. DNA has thymine, whereas RNA has uracil. So remember in RNA, A pairs with U, okay, instead of A pairing with T. DNA is double-stranded, while RNA is single-stranded. Now, if I show you the mark scheme to this question, going through the mark scheme, 6 marks, we need similarities and differences. There are 8 potential marking points. So first of all, similarities, it's a polymer of nucleotides. Except chain for polymer. Remember, I said it's a polymer made up of many repeating units known as nucleotides, and therefore that's what makes it a polynucleotide strand. The nucleotide has a pentose sugar, a pentose nitrogenous base or nitrogen containing organic base, and a phosphate group, which we also stated. We know that they all have cytosine, guanine, and adenine as the nitrogenous bases, and we picked up on these 2 points. The fourth point that we could have picked up on was that they both also have phosphodiester bonds. Okay, remember it's the strong covalent bond which is built between the phosphate of one nucleotide and the deoxyribose or the pentose sugar from the other nucleotide. Okay, it's the bond, the strong covalent bond that is formed between them two. Okay, in terms of the differences, we built on deoxyribose versus ribose, thymine versus uracil. You need to make it comparative, so you need some comparative phrase. In this mock scheme, they've just put V, but we've used whereas in most cases. We've used while over here, we've used whereas over here, and we used while over here. So you need a comparative phrase. You can't just say that one has this and that's it. You have to make a comparison. One has this, whereas, or while, or but the other one has this. You need to state what each one has, just so the examiner knows that you know what each one of them have, and you know you're making a comparative difference rather than just guessing what the answer is. Okay, one is long versus short. We never really stated that long versus short, but DNA is longer, whereas RNA is shorter. And then we've got DNA has a double helix, or DNA is double stranded, versus RNA, again, which is single stranded. Okay, and that would give you 6 marks for that answer. Okay, now the reason why I've laid it out as a table, it just makes it a bit easier for comparison. You could have just written a paragraph, but again, you want it easier for the examiner to read your writing and make out where you've gained the marks as well. So that's why I've just laid it out as a table there. Okay, if you guys have any questions, please let me know in the Q&A. I'll answer them, but I'm going to be moving on to the next question now. So it says, give the two types of molecule from which a ribosome is made. Okay, now we come across this later as well in a separate unit, but I did introduce it in this unit as well. So the two types of molecules a ribosome is made of is basically RNA. It's made up of RNA and a protein. Okay, now this is just something that you need to be aware of. Okay, RNA plus protein produces a ribosome. Okay, and here's the mark scheme: RNA, protein produces a ribosome. So that would get you 2 marks there. Okay, moving on. It says, describe the role of DNA polymerase in the semi-conservative replication of DNA. So we saw that DNA replication was semi-conservative, and we also saw that when we've got DNA strand like this, we saw that each strand will act as a template. So the strand will split and one strand will go like this, and one strand will go like this. Okay, so we've got the two separate strands of DNA, just like that. So we've got strand 1 and strand 2, and then we've got enzymes, we've got DNA helicase, and we've got DNA polymerase, and then we've got the free floating adjacent nucleotides, which are attracted to these template strands. So these, remember, are template strands of DNA. Okay, so then we know what there's normally exposed bases on each of these strands, like that. And then free floating nucleotides are attracted to the exposed bases, and then DNA polymerase catalyzes the formation of the phosphodiester bonds. Okay, so you've got free floating bases just in the middle, just like that. And remember, we saw that A pairs with T, and we know that C pairs with the G, adenine pairs with thymine, cytosine pairs with guanine, and these exposed bases will bind to their complement letter. And then what will happen is that the enzyme DNA polymerase will come on and it will catalyze the formation of the phosphodiester bonds, and it will produce a new double helix of DNA. Okay, now what we're getting over here is that each strand separates and acts as a template. Yeah, we don't get something like we've got a normal DNA strand like that, and then we don't form a new DNA strand from scratch. Okay, we normally have one strand which acts as a template. We don't just synthesize a new strand randomly. We need the DNA to be conserved. We need the DNA to be kept in place. We don't want alterations between the DNA, because then that would lead to genetic failure. It would lead to genes in your body being different at different parts of your body, and that's what we don't want. We want the DNA to be constant all through our body, and that's why we have the semi-conservative replication of DNA. Okay, so I've just summarized the process there. Okay, how do the strands break apart? We've got DNA helicase. DNA helicase breaks the adjacent hydrogen bonds, and then over here, catalyzing the formation of the phosphodiester bonds is DNA polymerase. Okay, and we've got the template strands of DNA, the free-floating nucleotides being attracted to the complement bases, and then DNA polymerase will finally fix together the bases, forming the phosphodiester bonds, and then you will have your brand new double helix of DNA. Okay, now it says, describe the role of DNA polymerase in the semi-conservative replication of DNA. Basically, what we've discussed there is that DNA polymerase joins together adjacent nucleotides. Okay, so I'm just going to write that down. So we've got DNA polymerase joins together adjacent DNA nucleotides. Okay. What else happens? It catalyzes the formation of what bonds? It catalyzes the formation of phosphodiester bonds. Okay, so it catalyzes the formation of phosphodiester bonds. Okay. And that's how I would answer this question. Okay, if we look at the mock scheme, joins adjacent DNA nucleotides, catalyzes condensation reactions, which we never stated, but it catalyzes the formations of phosphodiester bonds. Okay, so we've picked up 2 marks there. Now remember, the condensation, because of the condensation reactions, we form those phosphodiester bonds. Okay, remember, it's happening between the phosphate. How do phosphodiester bonds form? Let me just go over that. So we've got phosphodiester bonds. It forms between the phosphate of one nucleotide and the deoxyribose of another nucleotide. Okay, and this happens through a condensation reaction. So it happens via a condensation reaction. Okay, so it happens via condensation reaction, and each condensation reaction, a molecule of water is released. So a molecule of water is released in the process. Okay, so that's basically what happens when phosphodiester bonds are forming. Okay, moving on. So once we've looked at DNA replication, we've looked at DNA helicase, DNA polymerase. We then moved on to the Watson and Crick model of DNA replication. 2 scientists who investigated that DNA replication was semi-conservative and not conservative. So as in it was not fully resynthesized from scratch. Okay, so the question says, name the 2 scientists who proposed models of the chemical structure of DNA and DNA replication. Well, I've just stated this. It is Watson and Crick. Okay, so Watson and Crick were the 2 scientists who proposed the models of the chemical structure of DNA and DNA replication. Okay, remember, Michelson and Stahl were the people who proposed that DNA replication could be conservative or semi-conservative. Watson and Crick were the people that actually tested it out, and they brought up evidence to state that DNA replication was semi-conservative and not conservative. Okay, so we've got Watson and Crick there. It says a scientist replicated DNA in a test tube. To do this, he mixed an enzyme with identical single-stranded DNA fragments and a solution containing DNA nucleotides. Name the enzyme used in this DNA replication. Okay, so DNA is replicating. When DNA is replicating, there are only 2 possible enzymes that can be used in the process. So we've either got DNA helicase or we've got DNA polymerase. Okay, so the answer has to be one of these, DNA helicase or DNA polymerase. Okay, now can it be DNA helicase? Let's have a look. So replicated DNA, he mixed an enzyme with single-stranded DNA fragments. So the DNA fragments are already single-stranded. And there's a solution containing DNA nucleotides. Now, because the DNA fragments are already single-stranded, it has to be DNA polymerase. Okay, and I'll explain why in a minute. Okay, remember, DNA helicase breaks the hydrogen bonds between the complementary base pairs. Okay, that's what DNA helicase does. So remember, when we have our sort of DNA helicase, how does this unwind? Okay, DNA helicase makes the double helix unwind. Okay, it breaks the hydrogen bonds. So we've broken the hydrogen bonds and it unwinds the double helix to make it a single strand, a single helix. Okay, and then after that, that's all DNA helicase does. After that, DNA polymerase comes in and it reforms the double helix by catalyzing the condensation reactions. So catalyzes the formation of these phosphodiester bonds between adjacent nucleotides. So in this solution, we've only got single DNA fragments, which means that DNA helicase has already done its job. DNA helicase has made them from double helix to single helix already. Okay, the work's already done. They're already single-stranded. Now, because they're already single-stranded, all we need to do now is catalyze the formation to make the double helix structures back again. Okay, and the only enzyme, if we've crossed out DNA helicase, it has to be DNA polymerase. Okay, and that would get you 1 mark. Okay, so DNA polymerase is your answer there. Okay, moving on. It says, okay, now we're moving on to some questions on ATP, and then I think we've got a question on water, and then we're finishing off with a question on inorganic ions. Okay, so adenine triphosphate, ATP, is a nucleotide derivative. Contrast the structures of ATP and a nucleotide found in DNA to give 2 differences. Okay, guys, feel free to have a go at it yourself and check your understanding when I go through it with you guys. Okay, so we need to contrast the structures of ATP and a nucleotide found in DNA to give 2 differences. Okay, so first of all, a nucleotide found in DNA. What does that contain? Well, let's take a look now. We know that a DNA nucleotide contains deoxyribose. It contains 1 phosphate group. And it contains 1 nitrogenous base. In fact, if we were to just recap the structure of DNA and we were to draw the structure of DNA, then we would have our pentose sugar, which is our deoxyribose. We would have our nitrogenous base, and then we would have our phosphate group. Okay, so we've got our deoxyribose, which is our pentose sugar. We have our phosphate group, and then we have our nitrogenous base. Okay, that's just the structure of DNA. Okay, so we've looked at the nucleotide found in DNA. Now we need to look at the structure of ATP. Now remember, ATP is adenine triphosphate. Okay, so we're gonna have ATP. We're gonna have a ribose. We're gonna have we're gonna have a ribose. We're gonna have adenine, and we're gonna have 3 phosphate groups. Okay, so we know that ATP is made up of adenine. It's always adenine. That's the nitrogenous base that makes up ATP. We're going to have 3 phosphate groups. And the pentose sugar that we have is going to be ribose. Okay, so now we can make we can compare the structures. I literally listed everything that you need to compare. We just need to pick 2. Okay, so the first point that I'm going to say is the pentose sugar. So ATP we saw has ribose, whereas DNA nucleotide has deoxyribose. Okay, so that's the first thing. So okay, so ATP has ribose as the pentose sugar, whereas DNA nucleotide has deoxyribose as the pentose sugar. Okay. Now the second difference that I'm going to state, so we've said deoxyribose, so we know that ATP has 3 phosphate groups, whereas a DNA nucleotide only has 1 phosphate group, as you can see over here and over here. Okay, so we know that DNA nucleotide has only 1 phosphate group, whereas, again, you need to make comparison whenever you're looking at differences, whereas ATP has 3 phosphate groups. Okay, now if we look at the mock scheme, ATP has ribose and DNA nucleotide has deoxyribose. ATP has 3 phosphates and DNA nucleotide has 1 phosphate. So we said all that. Now the only thing we didn't say was, and there were 3 possible marking points over here. ATP base always adenine. You could have said that. And in DNA, nucleotide base can be different. It can vary. Okay, so that's another point that you could have said there for your second mark. Okay, but we've got 2 marks in that question. Okay, so moving on, we have another question on ATP. So it says, describe how an ATP molecule is formed from its component molecules. Okay, so this is an interesting question because it's worth 4 marks. You might think that there are not many points to talk about. Okay, but if we take this step by step, we need to see how an ATP molecule is formed from its component molecules. Okay, okay. So if we start off from the scratch, we know that ATP is made of ADP and 1 phosphate. So we've got ADP, adenine diphosphate, combines with a phosphate group to form ATP. And because ATP is being synthesized, you have ATP synthase. And in the process, it's a condensation reaction. So you're going to form a bond and then you're going to release a phosphate group in the process. Okay, so describe how an ATP molecule is formed from its component molecules. So if we start off, let's describe ATP. So ATP consists of ribose, 3 phosphate groups, and adenine. Okay, that's what ATP consists of. How is it formed? ADP reacts with a phosphate to produce ATP and water. This is through ATP synthase via, and remember in the other mock scheme we saw that it was a condensation reaction. So via a condensation reaction, releasing a molecule of water. Okay. Describe how an ATP molecule is formed from its component molecules. So its component molecules are ADP and PI. Okay. ATP synthase is the enzyme which catalyzes the reaction. Water is released. It's a condensation reaction that occurs in the process. Okay. From its component molecules, we've named all the component molecules. We've got ribose, 3 phosphate groups, and adenine. Okay, so I think, yeah, that's everything for this question. Obviously, it doesn't seem that scary once you've literally jotted down everything you know. So 1 and 2 accept 2 marks for the correct name of 3 components: adenine, ribose, and 3 phosphates. Ribose, 3 phosphates, adenine. So we've got 2 marks there. Condensation reaction, which we stated there, and ATP synthase, which we stated there. Okay, and that would be worth 4 marks. So normally you will see that with these questions, it's really picky. It's really hard to understand where the marks come from unless you've had a lot of practice. But again, it's just note down everything that's relevant to the question, and the marks will be in your answer, whatever you've noted down. Okay, so our penultimate question over here is on water. It says state and explain the property of water that helps to prevent temperature increase in a cell. Now, the property of water, remember there were 5 properties. So we had a metabolite, we had solvent, we had a high specific heat capacity, we had a high specific latent heat, and we had strong cohesion tension. Okay, now which of those 5 properties helps prevent temperature increase in a cell? So remember, a metabolite is the sum of the chemical reactions. Solvent makes it very useful for dissolving and also chemical reactions. High specific heat capacity allows a buffer in a change in temperature. Okay, and we're talking about temperature here, so the property is going to be a high specific heat capacity. So high specific heat capacity. Okay, that's what our property is. An explanation, how does it help prevent temperature increase within a cell? Well, if we think about it, it buffers a change in temperature. Okay, so the explanation is that it buffers changes in temperature. Okay, and remember, for specific heat capacity, I said just learn this statement over here. It's always applicable for all mark schemes. High specific heat capacity buffers changes in temperature. Okay, and here high specific heat capacity buffers changes in temperature. Same word to word. It's the same mark scheme that comes up over and over again. So this is basically a statement that you just learn. Okay, and that's worth 2 marks. And finally, guys, last question to summarize this unit. It's the last topic, which was inorganic ions. So it says describe the roles of iron ions, sodium ions, and phosphate ions in cells. Okay, so a lot of this you will come across in more practical situations later on in biology. Obviously, this is the second unit into biology, but a lot of these you will cover later on in biology as well. So, but we did look at some roles in terms of iron. If we start off with iron, iron binds with hemoglobin to transport oxygen. Okay, so it binds with hemoglobin to transport oxygen. Okay, so that's what that's what iron does. Okay, now sodium ions and phosphate ions, these have very sort of a lot of uses in cells. And if this is worth 5 marks, you're probably thinking that we may need to we may need to write down more than 1 use. Okay, and that's absolutely correct. So I will write down 2 uses of sodium ions and 2 uses of phosphate ions. Okay, so if I start off with sodium ions. What's the use of sodium ions? Well, we looked at the use of sodium ions. It's involved in the co-transport of glucose and amino acids. So co-transport of glucose and amino acids. We looked at this reason in our last lesson, but this is coming up in our next unit as well. So we're going to have an opportunity to have a look at it in more detail as well. Okay, and sodium ions, we didn't look at another reason, but I'm going to introduce another reason. Sodium ions, because it's an ion, it's actually really big. It affects the water potential. Okay, and we will come across this in our next few lessons as well. Okay, so it affects the water potential. And the reason why this, if I explain this now, imagine you're looking at osmosis, okay? And osmosis is the movement of water molecules from an area of high concentration to an area of low concentration through a partially permeable membrane, okay? So we've got osmosis occurring over here. Now, in this case over here, if we had big molecules like sodium present, it's going to affect how fast this water diffuses across by osmosis. Because there are big sodium ions which are in the way, it's going to affect the speed that osmosis is going to happen. It's going to affect the water potential. It's not going to be as high as expected. Okay, and that's what another role that sodium ions have. It's to affect the water potential. So at different places, different ions, water can be absorbed at different paces. Okay, phosphate ions. If we look at phosphate ions, now we've looked at phosphate so much, we've looked at it in this unit. Phosphate is used in ATP. Okay, phosphate is a component of ATP. Okay, it's a component of ATP. Phosphate is also used in nucleotides in the phosphodiester bonds. Okay, so what it does, it joins with deoxyribose in a phosphodiester bond. Okay, so that's what phosphate does. Okay, and again, phosphate is another ion. It's a big ion, so it can also affect the water potential. So you could have had that as a reason for phosphate as well. Okay, if I show you the mock scheme now, let's try get rid of. Okay, so hemoglobin binds or associates with hydrogen involved in the transport of oxygen. Okay, sodium ions co-transport of glucose amino acids into cells. Sodium moved out by active transport. Again, most of sodium ions uses are in the transport in cells, which is a whole unit by itself, which is in the next unit that we look at. Phosphate ions affects the water potential, nucleotides, ATP, phosphorylates other compounds. Remember, when phosphates are released, they can go and combine with other compounds such as glucose, and it can make them more reactive. They are hydrophilic, water soluble, part of the phospholipid bilayer. Again, something we will look at in the future.